As we look at today's focus, I'll be introducing Mr. Cephas, who will take you on glycolysis, basing the fat on scholar aspect of a man. So, hook on and enjoy glycolysis broken down to your best comprehensive understanding that you hardly forget. Glycolysis. This is the breakdown of glucose to form pyruvate. The pyruvate is then funneled into TCA-cycle. Objective of today's class: comprehensive knowledge of glycolytic pathway and clinical importance. Understand how to approach undergraduate exam as related to glycolysis, that is, undergraduate exam focus oriented. By the end of this class, we aim that you will not only comprehend glycolytic pathway, but you can easily relate to glycolytic pathway and apply them as relative to life. And at the end of this class, you'll be able to know how to get questions relative to glycolytic pathway, especially undergraduate questions, to pass your second MBBS exam. Content: diagrammatic illustration and explanations. Here we give you details, illustration of glycolysis. We give you details, explanation of glycolysis, and relative event that took place at a particular phase. Second content: glycolytic pathway and associated enzymes. Yes, we have a special design illustrations which helps it to retain within your memory so that you hardly forget. So there is no need cramming. Regulations of glycolysis. This is very, very paramount. So when you learn glycolysis, you know how to regulate it, how the body regulates it. So follow me by name, Mr. Cephas. Let's debunk glycolysis. First of all, before I proceed ahead, there are several glucose transporters across the cell membrane. Now, glucose, we know, is a large molecule which needs a transporter to enable it cross the phospholipid bilayer of a cell membrane. Now, basically, I'll be focusing on glucose transporters, four majorly: GLUT1, GLUT2, GLUT3, and GLUT4. So, why am I focusing on glucose transporter before proceeding to glycolysis? This is because before a glucose molecule can be broken down to pyruvate, just like we said earlier that glycolysis is breakdown of glucose molecule down to pyruvate, it has to be transported into the cells. So, my focus here is only basically on GLUT1 to 4. GLUT1, you can find it on blood cells. You can find GLUT1 on the surface of fetus cell membrane. You can find GLUT1 on blood-brain barrier. GLUT2, you can find GLUT2 in kidney, liver, pancreas. GLUT3, you can get GLUT3 placenta neurons, kidney. Then GLUT4, muscles and adipocytes. Now, how can you recall these glucose transporters and relative cells they are found? That's the reason why I came up with an acronym: BB, key, lips, blink, mother, father. So, wherever you remember this, it can help you to comprehend and not forget. But then note, GLUT1 to GLUT3, they are insulin independent. GLUT4 is insulin dependent. That is to say, GLUT4 requires, rather, they work hand in hand with insulin. Follow me and you will comprehend. So glycolysis, as I said, glycolysis is breakdown of glucose down to form pyruvate. and the pyruvate then channel down to TCA-cycle so as to generate reducing equivalent, which will now be channeled down into the electron transport chain to generate ATP. The sole aim of glycolysis for energy production from glucose. Note, glycolysis is different from glycogenolysis. Glycogenolysis is breaking down of glycogen stored in the liver to form glucose when the blood glucose falls low. So, here is diagrammatic representation of the explanation above. So here, we're going to take it very easy for comprehension's sake. Imagine this is a glucose molecule floating at the extracellular fluid. This is a glucose transporter, which if you can recall, we have GLUT1, GLUT2, GLUT3, GLUT4, as I am focusing on in this class. Then, these are enzymes within the cell. Note, glycolysis takes place in every part of the cell, body cells. So, it is not limited to only liver, just like gluconeogenesis. So, this glucose molecule enters through this glucose transporter. This is a cell membrane, and then when it gets in here, this is an enzyme. There are two enzymes who act synonymously. One, hexokinase, can be found in various tissues, or glucokinase, which is localized in the liver. This step is irreversible that this enzyme catalyzes. What happens when glucose molecules is being transported into the cell? Let's assume this is a liver cell or muscle cell. Hexokinase attaches phosphate group on the glucose molecule so as not to enable this glucose molecule to move backwards into the extracellular fluid. Thus, kinase reactions are irreversible, and kinase reactions involves ATP. Now, after then, the next step, as you can see here, there is another enzyme here, phosphohexokinase, phosphohexoisomerase. What does it do? Glucose phosphate being formed by the attachment of phosphate group by either hexokinase or glucokinase to glucose molecule is being isomerized. But hold on, I'm going to take it a bit easier so that you can comprehend. Good. Hexokinase is an enzyme that helps to phosphorylate. When you mean phosphorylate, you're adding phosphate group to phosphorylate glucose molecule so as to trap it within the cell, not to be able to move outward again. So glucose movement outward is inhibited once it's been phosphorylated. Good. Glucokinase is only in the liver, so someone can ask you what's the difference between hexokinase and glucokinase. Glucokinase is found only in the liver cell. Hexokinase are found in various body tissues. Please take note of this. Then, hexokinase can react or can phosphorylate little, even the minutest glucose in the body. But glucokinase is being stimulated into action, most specifically when there is high blood glucose. Then, after the phosphorylation, glucose molecule becomes offensive. Why do you have to add the phosphate group to me? I don't need it. And refuse to move to the next pathway. Then phosphohexose isomerizes this, changing the structure. This phosphate, calling glucose 6-phosphate, the 6-phosphate is simply indicating the fact that the phosphate group is being attached to position 6 of the carbon molecule. So then, the isomer of glucose 6-phosphate is then fructose 6-phosphate, which is then funneled down through the pathway. When this is done, another enzyme, phosphofructokinase, then comes. Recall that I said earlier that every kinase reaction are irreversible, and all kinase reaction involves ATP and ADP. So then, phosphofructokinase comes into the pathway with the utilization of ATP to add one phosphate group from ATP into the substrate and remove the remainder as ADP. So, once it adds another phosphate group, when you recall the structure of glucose or fructose, you can see there are 6 carbon molecules. Now, this is position 1, for instance, this is position 6. So once recall initially, we have fructose 6-phosphate. It comes in here to add phosphate group to position 1 of the glucose chain. So making it fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate. Once that is achieved, aldolase enzyme comes in to tear this at position 3 to form 2 different molecules. There's a cleavage to form this molecule and this molecule by an aldolase enzyme. This is known as dihydroxyacetone phosphate. Why this is glyceroaldehyde 3-phosphate? Please, this is phosphate, not glycerate. Kindly bear and correct. This is supposed to be glyceraldehyde 3-phosphate. So, but there is an important information you need to grab here. There's an isomerase enzyme. There's an isomerase enzyme here. Now, the only pathway, the only substrate that is funneled into glycolytic pathway at this junction is glyceraldehyde 3-phosphate. Recall I said this is phosphate, not glycerate. It's supposed to be phosphate. Good. But this isomerase enzyme, this can be a waste. It isomerizes dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. But there is a key instruction or information rather that you get to grab from here. At this point, glycerol can enter this pathway to form pyruvate. Once it enters this pathway, glycerol is 3 carbon molecule, enters this pathway forming dihydroxyacetone phosphate, then isomerized to glyceraldehyde 3-phosphate. The enzyme that catalyzes the reaction here is known as glycerol synthase. Let's go on. Then, the enzyme here is glycerol kinase, not synthase. Please take note. Glycerol kinase, not synthase. Thank you. Then here, this is another step Step 5 of glycolysis. Glyceraldehyde 3-phosphate dehydrogenase. A dehydrogenase enzyme. This is a dehydrogenase enzyme, supposed to be S. Dehydrogenase enzyme comes in to remove hydrogen ion. Once you hear dehydrogenase, hydrogen molecule is being cleaved off, and in process it's adding phosphate group to the pathway. So cleaving off hydrogen molecule and then adding phosphate. This step is very paramount because the NADH here and NAD plus is being utilized as step 10 of glycolytic pathway, whereby during anaerobic condition the body channels pyruvate instead of into acetyl-CoA, channels it into lactic acid formation catalyzed by lactate dehydrogenase enzyme. So once this is done, recall that at this point this molecule is moving towards this direction. So we are working with 2 molecules here instead of 1. When this is achieved, this is the reason behind you are seeing 2 here. 2 molecules here. So 2, 2, 2, everything we'll be doing henceforth is 2 by 2. Is that interesting? Okay. So once that is done, what happens? If you look out here, glyceraldehyde 3-phosphate is telling you that it's only 1 phosphate being attached at position 3. However, as glyceraldehyde 3-phosphate dehydrogenase enzyme adds 1 phosphate again to position 1, making it 1,3-bisphosphoglycerate. Recall, it's occurring in 2. Good. So 1,3-bisphosphoglycerate. Once that is being formed, a kinase enzyme comes in. But note, this step is extremely paramount. Recall that I said kinase reactions are irreversible. Irreversibility of kinase reaction is dependent on the pathway following the reversibility. What do I mean? Kinase reactions cannot be reversed following the same pathway. However, it can be reversed, it can be circumvented using another pathway. For instance, the pathway to circumvent glycolytic irreversible steps are gluconeogenic pathways. Good. However, here, this kinase enzyme is reversible. Please take note. In glycolytic pathway, all kinase enzymes, all kinase reactions are irreversible except step 6 of glycolysis. And note another important thing here. Here, it's known as substrate level phosphorylation. I will explain. Recall that glyceraldehyde 3-phosphate dehydrogenase enzyme added phosphate group to the pathway, forming this. Now, previously we've been making use of ATP to give off ADP, but currently ADP is being utilized to cleave off ATP. This is the energy currency of the body. This is the energy the body requires. So, looking backward, here, let me wipe my board so that it can be clearer. Now, here, this NADH is a substrate, rather is a reducing equivalent that can be channeled through electron transport chain to generate ATP. So you can see it has to follow another procedure so as to generate ATP. But in this case, ATP is being generated at this point. Rather than utilization of ATP as seen in the previous slide, just like here, utilization of ATP, here then ATP is being produced. So that's why it's called substrate level phosphorylation. In other words, for more comprehensive reason, you can see that ATP, the phosphate is being added to the rest, but at this point, this is a substrate we are working on. Coming down here, the phosphate at this point, point 1, is being cleaved off. So, substrate level phosphorylation. So once that is done, then the name changes depending on the carbon number that the phosphate group is being attached to 3-phosphoglycerate. Then once 3-phosphoglycerate is being formed, a mutase enzyme comes in. We all know that mutation mostly doesn't give rise to positive things. It gives rise to alteration of original thing or original molecule. So looking at here is 3-phosphoglycerate because phosphate group is being attached to position 3. But once mutation comes, it cleaves off this phosphate group and attaches it as position 2, making it 2-phosphoglycerate by this enzyme. As you can see, the enzyme is looking ugly. It's coming to alter the genetic makeup of this glucose molecule. Good. Now, what's next? Once that is done, there is an enzyme that comes in, which is an enolase enzyme. It comes in here to catalyze the reaction, the conversion of this phosphate, 2-glycerophosphate, into another molecule. Please note that the sole aim of glycolytic pathway is to produce pyruvate, which can be channeled into TCA-cycle so as to generate reducing equivalent, which can be channeled then into electron transport chain to produce it. So pyruvate, which is our targeting product, does not have phosphate group attached to it. So therefore, this pathway, enzymes involved mobilize themselves to ensure that phosphate is being cleaved off from the 3-carbon molecule so as to produce pyruvate. Then, what happens? Enolase enzyme comes. It doesn't cleave off the phosphate group. Rather, it weakens the phosphate group so that a stronger enzyme can be able to cleave it off. So, once it weakens it, it is called phosphoenol pyruvate. Phosphoenol pyruvate is not the substrate. Good. But I would want you to listen carefully here. This step of glycolysis, step 8, is of great importance. Why? Medically, fluoride is an allosteric inhibitor of enolase enzyme. So, this magnesium is here to prevent the binding of fluoride to enolase enzyme so as to ensure the pathway continues. Now, as a medical lab scientist, you are given a blood sample of a patient. You are meant to check the blood glucose level. The first thing that comes to your mind is to add fluoride to the glucose sample. Why? Without adding fluoride, there will be no inhibition at this point. The glucose will keep on degrading. By the time you get to the lab, the original value of the patient's blood glucose level has reduced, and then after your lab analysis, getting back results to the medical doctor or the physician, you are bringing false results, which you know can be disastrous. So please take note. Ensure that you add fluoride to glucose blood sample. So once phosphoenol pyruvate is being formed, then kinase enzyme comes in again. Pyruvate kinase, with the aid of ADP, produce ATP. Recall, at step 6, ATP was produced. At step 9, ATP was produced. Thus, both steps are regarded as substrate level phosphorylation. Then, once pyruvate is being formed by pyruvate kinase, this is the pyruvate being formed. Recall step 6 and 9 as substrate level phosphorylation. Once pyruvate is being formed, then pyruvate is 3-carbon molecule. All the enzyme did was to cleave off the weakened phosphate group so as to form 3-carbon molecule having no phosphate group. Under aerobic condition, pyruvate is being converted into 2-carbon molecule known as acetyl-CoA. Acetyl-CoA is a substrate to TCA-cycle. Acetyl-CoA and the TCA-cycle, in our next class, when we walk towards TCA-cycle, you get to know that acetyl-CoA combines with oxaloacetate to form citrate, which then generate enough energy to drive the living being. But this is catalyzed by a complex enzyme, pyruvate dehydrogenase complex enzyme, adding CoH. This is HS. Okay, just try, bear with us. Alright, so guys, next, under anaerobic condition, mostly seen in athletics, the pyruvate is being channeled into 3-carbon molecule lactic acid, catalyzed by an enzyme, lactate dehydrogenase. You can see this is step 10. The NADH and NAD plus from step 5 is then utilized. So, too much accumulation of lactic acid gives rise to acidic blood. Now, checking blood sample. Once there is a high level of lactate dehydrogenase in blood sample, is an indication that there is a hepatic problem. So, take not for granted. A recap. From step 1, once glucose is being intracellularly placed, phosphate group is being attached to it so as to prevent its going back. Then, step 1 is irreversible. Step 3 is irreversible. Step 6 is irreversible, known as substrate level. Step 6 is reversible, catalyzed by kinase reaction, kinase enzyme, known as substrate level phosphorylation. Recall, all kinase reactions are irreversible in glycolytic pathway except Step 6. Step 9 is irreversible. It's a kinase reaction. Now, here, conversion of pyruvate conversion to acetyl-CoA is irreversible reaction because carbon molecule was cleaved off, forming 2. So to reverse it is pyruvate carboxylase that comes in to add one more carbon molecule here and then push it backward to form pyruvate. That is circumvented through another pathway. Likewise, this reaction catalyzed by pyruvate carboxylase, to enable it to be converted back, that means carbon molecule must be cleaved off. But during gluconeogenesis, you will realize that oxaloacetate cannot permeate through the mitochondrial cell membrane, but it can be converted. It can be isomerized to malate outside the mitochondria, then be transformed back to oxaloacetate, then channeled back to phosphoenol pyruvate. All this is being catalyzed by an enzyme, phosphoenol carboxykinase enzyme. Okay. Summarily, kinase reactions are step 1, step 3, and step 1, step 3, and step 9. They are all irreversible. Recall, the whole kinase reactions in glycolytic pathway are 4. Step 1, step 3, step 6, and 9. However, step 6 is reversible. Step 1, 3, and 9 are irreversible. 2. Step 6 and 9 are substrate level phosphorylation. These are exam focus. Please pay attention. Reversibility is via gluconeogenesis. Via gluconeogenesis, glucose 6-phosphatase, domicile in the smooth endoplasmic reticulum of any cell, converts glucose 6-phosphatase into glucose by cleaving off the glucose, the phosphate group attached to the glucose. When we get to gluconeogenesis, we get to know pyruvate carboxylase, step 9, is also just as illustrated here. Pyruvate carboxylate adds one more carbon molecule here to produce pyruvate. Fructose 1,6-bisphosphatase cleaves off the phosphate group to form fructose 6-phosphate. Note this, your examiner can ask you a question, especially in medical school and as undergraduate. Bisphosphate and biphosphate, what's the difference? Once we have carbon molecules separated by each other bearing phosphate group, it's known as bisphosphate. Carbon molecule close to each other bearing phosphate group is known as biphosphate. Example. When you get to this point, as you can see here, you have phosphate here, you have another phosphate here. There is a carbon molecule separating the two. It can be 1, it can be 2, it can be more. This is bisphosphate. But assuming there is a phosphate group here and another phosphate group here, it is called biphosphate. And then if you have phosphate group here, and phosphate, you can call triphosphate. But just know this, that in bisphosphate, there is a carbon molecule separating, at least one carbon molecule separating the carbon molecules bearing the phosphate group. While biphosphate, there is no carbon molecule separation. Fate of glycolysis in RBC. Please take note. There is no mitochondria in matured red blood cells, so therefore the only source of energy is glycolysis, where the end product is lactic acid. When energy is trapped by oxidation, That is, oxidation implies breakdown of reducing equivalents such as NADH. It is known as oxidative phosphorylation. Summary: glycolysis, either glucokinase or fructokinase, makes use of 2-ATP. Anaerobic process, anaerobic glycolysis. End product is lactic acid. Glycolysis takes place in the cytoplasm. These are objective questions in medical MBBS, second MBBS exam. Glycolysis takes place in cytoplasm. TCA-cycle occurs in mitochondrial matrix. Oxidative metabolism occurs in the internal fold of mitochondrial membrane. Assessment: you can do well to assess yourself from this. You can as well go to the site and get these materials and assess yourself. The following are the sites of glycolysis except one: mitochondria, liver, muscle. None of the above. Of course, the answer is none of the above, because glycolysis, we said earlier, occurs in all body tissues. State the kinase reactions of glycolytic pathway and enzymes involved. As it talked above, kinase reactions, you have step 1, step 3, step 6, and step 9. Step 1, hexokinase or glucokinase. Step 3, phosphofructokinase. Step 6, you have step 6 is kinase enzyme. Step 9, also kinase, pyruvate kinase. State the steps: substrate level phosphorylation, as we said, step 6 and 9. So go ahead and have access to the assessment and assess yourself. Assessment Session B. This is very critical and paramount to us. Please, how was the lecture? Did you enjoy the class? Do you love MELVISWORLD's teaching approach? Do you want to get more explanations from MELVISWORLD? List the difficult areas you would want MELVISWORLD to assist you on. Kindly state areas of improvement you expect from MELVISWORLD. Please email us at mmelvisworld@gmail.com.